Hello dear learners, today we are going to learn how to visualize the active site of a receptor. For this we can take help of the literature or visualize the binding site of the inhibitor attached to the receptor. We will use the PyMOL software for this. We open the receptor 6LU7 previously downloaded from PDB. So this is the 6LU7 structure. The red dots indicate water molecules. We have to remove them for better visualization. Then we need to analyze the sequence of amino acid and find out the odd one out in the sequence. Select it, it will be selected in the structure. That is our inhibitor. Once the inhibitor is selected, we can change the color of the amino acids to one color and color the inhibitor to another color for better visualization. Here the inhibitor is colored with cyan. We can rename our inhibitor. Next step is to visualize the interactions of the inhibitor with the nearby amino acids. We select the amino acids which are in interaction with the inhibitor for the purpose of labeling. We rename our selected amino acids. Now we hide the entire sequence of amino acid of the receptor and visualize only the inhibitor and the interacting amino acids. Next step is to label the interacting amino acids and note them down for the subsequent steps of docking. We can also change the background and take high-quality images for publication purpose. Here the amino acids which are found to be interacting includes phenylalanine at 140, glycine at 143, histidine at 163, 164 and glutamine at 166.